And for today's report, we're going to tackle about the changes in chromosome structure or chromosome abbreviations.
History of chromosomes. The number of human chromosomes was published in 1923 by Theophilus Painter. By inspection through the microscope, he counted 24 pairs, which would mean 48 chromosomes. His error was copied by others, and it was not until 1956 that the true number 46 was determined by Indonesian-born cytogeneticist NT2, who discovered the structure of chromosome. Walter Fleming. He is the first biologist scientist who discovered chromosome in the late 1800s. After the light microscope was invented, he observed many tightly wound, elongated structure in cell nuclei. Later it was found that chromosomes are made from DNA, the cell's genetic materials. Father of chromosome. Hermann Hinking, he was the German biologist, noted that the X chromosome was special in 1890 in Leipzig. Hinking was studying the testicle of Pyrochoris and noticed that one chromosome did not take part in meiosis. Chromosomes are so named because their ability to take up staining. Chroma in Greek means color.
How the chromosome structure changes. So the changes in chromosome structure can come about through deletions or deficiency, duplications, and rearrangements. So loss of a segment of the chromosome is known as deletions, whereas addition of a segment of chromosome on the same or different chromosome is known as duplication. So it means if loss of a segment, it is a deletion, but if addition of a segment, it is a duplication. So there are four main types of structural chromosomal aberrations. So number one, it is deletion, duplication, inversion, and translocation. So the first major types of chromosomal aberration is the deletion. This occurs when a portion of the chromosome is deleted or taken out, which can make the chromosome less functional. For example, when part of a short arm in chromosome 5 is deleted, this causes Cri-du-Chat syndrome, common symptoms of which are reduced head size and high-pitched crying in infants. So this is the example of the deletion. So look at the picture, the Cri-du-Chat syndrome, or the 5p minus syndrome. So it is one example of genetic syndrome caused by a deletion where part of the number 5 chromosome is missing or deleted. So this is the second major types of chromosomal aberration, the duplication. So it is a part of the chromosome is duplicated, resulting in extra genetic materials. This occurs in Charcot-Marie tooth disease type 1, which duplicates part of chromosome 17, causing muscle weakness. So it is the one example of duplication. So one example of a rare genetic disorder or duplication is called the Pallister-Killian syndrome, where part of the number 12 chromosome is duplicated. Pallister-Killian syndrome is a chromosomal disorder caused by having an extra chromosome or duplicate chromosome. So it is a third major types of chromosomal aberrations. So the inversion. So a chromosome happens when the genetics materials is inverted or flipped in the opposite direction. Inversion do not often result in disease and most commonly affect chromosome 2. So the one well-known example of inversion is the hemophilia, a disease that inhibits the ability of blood to clot. Researchers discovered the gene responsible for hemophilia years ago, but also found that DNA from a person with hemophilia had the same genetic makeup as a person without the disease. So the last major types of chromosomal aberration is the translocation. So it occurs when a piece of one chromosome has broken off from its original location attached to another chromosome. So the most common example is a Robertsonian translocation, which results when two acrocentric chromosomes or chromosome width of unequal length due to an uncentered centromere, lost the short arms of the chromosomes, and the two long arms consequently conjoin. Robertsonian translocation are one potential cause of trisomies. So it is an example of translocation. The translocation in this example is between chromosomes 14 and 21. When a baby is born with this type of translocation chromosome, in addition to one normal 14 and two normal 21 chromosomes, the baby will have Down syndrome. So this is also called the translocation Down syndrome. So what is chromosome structure and function? Chromosomes are one thread-like structure present in the nucleus which carries genetics information from one generation to another generation. They play a vital role in cell division, heredity, variation, mutation, repair, and regeneration. So why is the structure of chromosomes important? So without such packaging, DNA molecules would be too long to fit inside cells. So the unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called the histones. So that's why the structure of chromosomes is very important. So that is our report about changes in chromosome structure or chromosome aberrations. Thank you, sir, and God bless.